This is a brief explanation of translation, the second part of the central dogma for general biology at RCBC. We already talked about transcription, which was using DNA to make RNA. Now we want to discuss how RNA is translated, the language of nucleic acids are translated into the language of amino acids to build a protein. What we use here are three-letter words called codons, and those codons will direct a specific tRNA molecule to bring in a specific amino acid. So we have a codon, which is a part of the mRNA. We have an anticodon, which is part of tRNA. And the tRNA, take a little closer look at tRNA, is a single-stranded piece of RNA that folds on itself, and this indicates we have some complementary base pairing here, hydrogen bonding, that makes it this clover leaf structure. Now, here is the anticodon, which you just saw. That's on one end, and the other end, the 3' end, will hold the amino acid. So an anticodon dictates a particular amino acid that will be held by that tRNA. The other player in translation is the ribosome. And the ribosome has two different units in it. And we'll see that in this slide here, perhaps a little bit better. It has a large subunit and a small subunit. Also knowing here that it's made up of many different proteins and also rRNA. It's called ribosomal RNA. There are 3 parts to this ribosome. And what you can see is that the ribosome is going to hold or sandwich the mRNA. And these 3 parts that I told you about in a previous lecture are called EPA sites, but that's where the tRNAs come in and where the codon matches the anticodon and a particular amino acid is added to the long chain of protein. Let's take a look at how this is initiated, how elongation occurs, and then how it all ends. So with initiation, here you can see we have that small subunit which attaches to the mRNA and holds on to the mRNA. We can see that the first codon, the start codon, is AUG, and the very first tRNA comes in and base pairs with the codon. That start codon, there is only one. It always holds a methionine, so we always begin translation with a methionine. Once this happens, the larger subunit sits down, and that is initiation of translation. Elongation is now we will have the tRNA will bond with the codon, and then the second tRNA comes in that matches the codon. Eventually, this tRNA will pass the growing strand of amino acids onto this tRNA, and then he will move out of the ribosome altogether. So elongation is just moving through. And I'd like to bring you back up to what we looked at earlier. Elongation is just moving through, read this codon, then read that codon, bring in the appropriate amino acids. Now, termination of translation is based upon the stop codons. So what we can see here is we have a stop codon. There is no tRNA that matches that stop codon. So a release factor sits in there, and then everything falls apart. there are 3 stop codons. You don't need to memorize those, but if we take a look here at the genetic code shows us all the different possible codons and which amino acids those codons will call for. Here's the methionine, the start codon. I told you there's only one. And then here are the 3 stop codons. So when the ribosome gets to one of those 3 stop codons, there's only a release factor that can come in and all of the translational machinery falls apart. Thank you.